Gina Gulucci, who also works with me on endometrial cancer research. And Gina came to me in the fall of 2016 to work on her research, so she's been with me for about 2 semesters. And she quickly put together a great grant and got it funded by, again, 2 separate funding agencies, both the university and also the Tri-Beta National Honor Society, and this allowed her to pursue her research. And I've known Gina for many years, and she is all over the campus, and many of you probably know her as Tri-Beta president and a tutor in the CAE and involved in many organizations on campus. So she somehow found time to also do research with me, and she probably sees me more than she would like to on a daily basis between all our different endeavors together. But I was very impressed with Gina because she kind of found this passion for research recently, and she's really flourished and grown a lot in these last few months. And she's really accomplished a lot in a short time. And she's currently finding her passion in terms of what is next for her after graduation. But she certainly is very adept at research, and she's a good critical thinker and problem solver, just like Safa is as well. And they work extremely well together as a team, and their research has really complemented each other. So she's going to be talking to you also about endometrial cancer, but she's looking at a slightly different aspect of this, kind of ties into Safa's research as an extension where Gina's going to be looking at perhaps reclassifying endometrial cancer in regards to epithelial to mesenchymal transition as opposed to estrogen receptor status based on some very interesting data that she obtained. And she will also be presenting at the Tri-Beta conference at the end of the month, as well as a poster at the STEMCon conference also later this month. So her April will be quite busy. So without further ado, I'd like to introduce Gina Gulucci.
Okay, so as Dr. Reed graciously introduced me, I'm Gina, and my project is called Using Epithelial Mesenchymal Transition as a Model for Clinically Characterizing Endometrial Cancer. To first understand my project, we have to know what is cancer. Cancer is the uncontrollable division of abnormal cells in the body, and as you can see, as the cells divide, a tumor starts to form. In this picture, the tumor is still above the basement membrane, but if it were to break through this basement membrane, the cancer would be considered invasive. About 12.7 million people are diagnosed with cancer every year, and about half of these people die from their diagnosis of cancer. I specifically looked at endometrial cancer, and endometrial cancer, as Safa explained before, is the uncontrollable division of the abnormal cells in the uterus. And endometrial cancer is the 4th most commonly diagnosed cancer in women, and about 13,000 women are diagnosed with endometrial cancer every year. Currently, endometrial is classified like breast cancer, and there's type 1 and type 2 breast cancers. Type 1 has an estrogen receptor, and type 2 does not, meaning that type 1 would respond to estrogen treatments. So this image shows that about 66% of breast cancers are type 1, whereas about 18% of breast cancers are type 2. So endometrial cancer is classified the same way. So basically, type 1 will respond to the estrogen treatments and will have a better prognosis, whereas type 2 endometrial cancer does not have an estrogen receptor and is not likely to respond to estrogen treatments, meaning it would have a poorer prognosis and it is more likely to metastasize throughout the body. To explain metastasis, metastasis is when the tumor has broken through the basement membrane that I was showing you before and enters the capillaries in order to travel throughout the body and basically create a new tumor in a new part of the body. So as you can see in this image, an early stage cancer has a tumor forming. And as it progresses, the tumor gets larger and it pushes against this basement membrane. If it breaks through the basement membrane, it can travel to a new area, and in this case, it's spread to the ovary. It has been hypothesized that epithelial mesenchymal transition is the driving force for metastasis, and metastasis is the leading cause for mortality in cancer. Epithelial mesenchymal transition is when you have your cells go from an epithelial state to a mesenchymal state. And what this means is that when the cells are epithelial, they look square-like, and they're very adhesive, and they attach to each other, meaning they're very stable to that basement membrane. When they undergo this process, they become an irregular shape, and showing that they lost their adhesion, which makes them more likely to break through that basement membrane and travel throughout the body. There are certain markers associated with this epithelial characteristic and this mesenchymal characteristic, and this project looked greatly into those markers in order to see if we can determine when these cells are in the epithelial state versus this mesenchymal state. So just to bring this back, currently endometrial cancer is classified in type 1 versus type 2, type 1 having that estrogen receptor and type 2 not having that estrogen receptor. But, as we saw in Sophia's research, some type 2 endometrial cancers have actually responded to estrogen, so this led me to believe that maybe we can look at these epithelial markers and the mesenchymal markers to change the way we clinically classify endometrial cancer. So this led me to my hypothesis that type 1 endometrial cancer cell lines will express high levels of epithelial markers, and that type 2 endometrial cancer cell lines will express high levels of mesenchymal markers and EMT-related transcription factors. The driving force behind this was because, as I was showing you before, cancers that are in a higher grade will have the should have the mesenchymal phenotype, so that's why we predicted this. First, I wanted to go over a few of my cell lines that I used. I used these 3 endometrial cancer cell lines. I don't know, I think if I stand over here, it won't make noise. I don't know. But I used RL952, which is classified as a type 1 endometrial cancer cell line, which would be my pre-EMT model. I don't know if I got it. Did you fix it? Yeah. Okay, thanks. So I used RL952 as my type 1 model for endometrial cancer, and that would be my pre-EMT model, meaning that we would predict that it has that epithelial phenotype that I was talking about before. My Ishikawa cells are my other endometrial cancer cell line. That would be my in-transition cell line. We would expect them to show epithelial markers and mesenchymal markers because they're switching in that EMT process. KLE cells have been classified to be a type 2 endometrial cancer cell line. So they would be my post-EMT model, meaning that they would have that mesenchymal phenotype if our hypothesis is correct. The first experiment that I conducted was called immunofluorescence. Immunofluorescence is when you take an antibody and you put it on the cells for a certain protein, or in this case we were looking for certain EMT markers. If that EMT marker is present, the antibody would attach to the antigen, and then we applied a secondary antibody that would allow that to glow so we could actually visualize the EMT marker in the cell. I first conducted a control experiment using 2 breast cancer cell lines with an EMT marker called vimentin. Vimentin has been shown to be higher expressed in a mesenchymal phenotype, and it has been shown that vimentin has been higher expressed in MCF7M cells because they are that type 2 breast cancer cell line that would have that mesenchymal phenotype. So as you can see in this experiment, the type 1, which is our MCF7M cells, have very little red. The red represents the vimentin and the blue represents the cell nuclei. So if you see around the cell nucleus, we have a little bit of red showing that there is some vimentin present. But in the MCF7M cells, there's a lot of red present in these cells, showing that vimentin is higher expressed in this cell line as predicted, which means that it can be correlated with that mesenchymal phenotype. So this led us to do an experiment in our endometrial cancer cell lines. So next we performed the same experiment using this vimentin in the RL952s, which is my type 1 endometrial cancer cell line, and my type 2 KLE endometrial cancer cell line. As you can see in the RL952s, there's barely any red present. And in the KLEs, there's a lot more red present around the nucleus. So this shows that the vimentin is higher expressed in the type 2 cancer cell line, and it also could be correlated to the fact that the type 2 KLEs do have that mesenchymal phenotype. The next experiment we did was a similar experiment called a Western blot. A Western blot also looks at antibodies and looking at specific EMT markers in the cells. To conduct a Western blot, you take the protein out of the cell and you separate it on a gel, and then you put it onto a membrane where we can then apply the antibody to be able to visualize it. So first we do a primary antibody to hook up to where if that EMT marker is present, and then the secondary antibody to actually be able to visualize this on the membrane. I did 4 Western blots, and I used the same one that I used in my immunofluorescence, my Vimentin. The letters at the top represent my 3 cell lines, my Ishikawas, my RL952s, and my KLEs. Just like the immunofluorescence, the Vimentin was higher expressed in my KLE cell line, as we can't see any bands in the RL952s. And there is also a band in the Ishikawa cells, because we would expect that the Ishikawa cells would show both epithelial markers and mesenchymal markers. It is accurate that Vimentin would be expressed in these cells because they are in transition going from epithelial to mesenchymal. So this supports my hypothesis. I also looked at E-cadherin, which is an EMT marker associated with adhesion. So we would expect this marker to be higher expressed in my type 1 endometrial cancer cell line if they are expressing the epithelial characteristic, because those were those nice square-shaped cells that were nicely packed next to each other, whereas the mesenchymal can move more freely. So as you can see in this picture, the bands in the Ishikawas and RL952s are pretty strong, which is what would be expected, especially for the RL952s if they are expressing that epithelial characteristic. And in the KLEs, there's a slight decrease. So this would be accurate to support my hypothesis, because we would expect the E-cadherin to be downregulated in my type 2 endometrial cancer cells. My next experiment I did was with another mesenchymal marker, which is called snail. This one's a little difficult to see, but if you can see, there are 2 bands in my Ishikawas and 2 bands in my KLE cell lines. Because snail is also a mesenchymal marker, we would expect to see it upregulated in the KLE cells, and it's also possible that it would be upregulated in the Ishikawas because those are my in-transition cell line. My last experiment was using beta-actin, which is a control. We would expect to see beta-actin just evenly distributed throughout all cell lines, and this, as you can see, they're all about the same intensity, meaning that we can use the results from this experiment and that this is basically conclusive. My last experiment that I did was called a wound healing assay. And when you do a wound healing assay, you take your cells and you put them in a dish, and you allow them to grow until they're confluent, meaning they cover the whole bottom of the dish. You then take a pipette tip and create a canal in the center of the dish, or a wound, and you see how quickly that canal is filled over a time period, and the cells that kind of fill that canal more quickly are going to be considered a cell line that is more invasive. In this experiment, we expected our type 2 cell line to be more invasive because we expected them to show that mesenchymal phenotype, meaning that they would be more likely to travel throughout the body. So these were my results from my wound healing assay in the endometrial cancer cell lines. The first pictures represent when I first made the canal, so T0. The second picture was after the first day, and as you can see, both canals are basically the same distance apart still. There hasn't been much of a difference. But in the last picture, after 36 hours, the RL952s, which are my type 1 endometrial cancer cell line, have filled up the gap a little bit. But if you look at the KLEs, they have significantly filled the gap, meaning that they may possibly be more invasive and express that mesenchymal phenotype that we did expect, that we predicted for our type 2 cell line. So what we could conclude from this was that each cell type expressed the predicted phenotype. As I showed before, the Vimentin and the snail, which were my mesenchymal markers, we did predict that they would be upregulated in my type 2 cell lines, and the results corresponded with that. The results may also support the development of a model for examining EMT and endometrial cancer. And this also shows that endometrial cancer could possibly be classified by EMT markers instead of estrogen receptor status, since it has been shown that type 2 endometrial cancers have responded to estrogen treatments. Some limitations of this experiment are that we use cell lines instead of patient tumors. If we were able to, we would like to use a 3D cell culture where we could have a tumor environment to be able to conduct the same type of experiments and see if we would get the same results. We also only have one good cell line for each phase of EMT. It would be more ideal to have multiple cell lines to look at to say that this data is actually conclusive for endometrial cancer. This study also only shows correlation and not causation. And what I mean by this is that vimentin, it was correlated to be high upregulated in a type 2 cancer with the mesenchymal phenotype, but we're not showing what causes that upregulation, and that would be a future direction to look more into that. And also, only a few genes were observed, and we would like to look at other genes for EMT in the future, and a couple of those genes would be TWIST and ZEB, which are also other mesenchymal markers that we could look at. I would like to acknowledge the USJ Provost Student Research Grant and Tri Beta for helping me fund my research, and Dr. Reed for helping me with all of my experiments and teaching me everything that I know, and Safa Saleh for being a great lab mate and helping me with some of my experiments. And that's it. Thank you.